This is really our last session, and our next and last invited speaker is Dr. Nancy Amato. She's an amazingly energetic woman. I'm just impressed by the number of things that Nancy is doing at all times. So first of all, let me tell you that she is the Okal, did I pronounce that correctly? Professor and interim department head of the Department of Computer Science and Engineering at Texas A&M University, where she also co-directs the Parasol Lab. She's no stranger to this area because she did her undergraduate degree at Stanford in mathematical sciences and economics. She got her master's degree at Berkeley, and then she has a PhD degree from the University of Illinois at Urbana-Champaign. Her main research areas are motion planning and robotics, computational biology and geometry, and parallel and distributed computing. In motion planning, she has made fundamental contributions to the development of sampling-based motion planning techniques, and actually she was very brave and looked at the cases that were the hardest of all, involving many degrees of freedom and narrow passages, but she also looked at deformable objects and planning for many robots. More than 10 years ago, she started using motion planning techniques for protein folding, and at the time, nobody expected that she would be successful, but she persisted, and she is extremely successful, and I hope she's going to tell us about this. Besides her technical contribution, I want to say that I can hardly think of people who have contributed so much to our community. And Nancy has been on the conference paper review board for IROS, has been at the editorial board of IEEE transactions on robotics and automation, and also transactions on parallel and distributed computing. She has served the community in many, many different roles, from invited speaker to administrative committees, and also she has had a very, very active role in outreach and the activities that have impacted many women students and underrepresented minorities. In particular, I would like to note her involvement with NC-WIT, and also CRAW, and the CDC, which is the Coalition to Diversity Computing. She has numerous awards. I'm not going to take the time to go through them, including teaching awards at Texas A&M University. Please join me in welcoming Dr. Amato.
Thank you, Lydia. Nice set. So that was a very generous introduction, and coming from someone who herself is probably one of the most involved and indefatigable persons in the community. So today what I'd like to talk to you a little bit about is motion planning and how we can use it to study molecular motions. So most of us in this community here, I hope, know what motion planning is, but kind of in its most basic form, we're given some movable object, a description of your environment, and you want to find a sequence of valid configurations that will take your movable object from the start to the goal configuration. And what is showing here is this alpha puzzle problem. I don't know how many of you played with it, but I have a couple physical ones that you can pass around and play with, but these are basically twisted nails. And this is what is known as a very challenging narrow passage problem. Once you know the trick, which is you need to know essentially that configuration right there, then you can separate them. But if you're just kind of randomly trying to discover this, it's very difficult to do so, because it's a very small subset of the configurations that are crucial. Now there's many other applications of motion planning that are outside of robotics. I won't go over the robotics applications here because we're all very familiar with them. But here's just, excuse me, some examples. On the upper left is an example from a mechanical design, a CAD system. This was an example from GE where they actually, the engineer designed this part, but they chose not to actually manufacture it because they weren't able to determine a priori that it could actually be assembled. They knew that the two parts fit together in their final configuration, but they didn't know it would be possible to actually put them together. It was with a motion planning algorithm that we were able to determine that they could have in fact gone with that original design. On the upper right, there are people evacuating a building. Oh, it just stopped now, but they were leaving the building, going into their cars, and then leaving. On the bottom right here, we have these deformable objects. Here we have the teapot and the ducky, and they're both following the same trajectory, and they're deforming to avoid collision with the surrounding objects. This would have applications, this one in animation, but there are also applications in things like surgery. And then on the bottom, there's a puzzle that has many different parts, and it's essentially a disassembly puzzle. All of these problems here and all these animations that I've shown you are solved using our motion planner, which is based on the probabilistic roadmap method that was pioneered by Lydia in her doctoral work. So these are just examples of motion planning and these sampling-based methods that have been applied outside of robotics. And what I want to do now is kind of give you some motivation about why we might think we could apply these methods to a molecule. So the way I got into it is actually, I think it makes a good story. Someone who I won't name, who's here today, visited Texas A&M, and he showed an animation or a movie of a robot folding a box. And I asked him, you know, how did you come up with the plan for folding that box? And I asked if he had used a probabilistic roadmap method, and he said no, he didn't. And yeah, we disagree today whether or not he kind of implied that he didn't think a probabilistic roadmap method could do it or not. But that was my impression. And I was sitting there with one of my students, and I suggested to my student that we should try that. And then basically the next day we had an animation of not this periscope here, but a simple box folding. And then one day I was visiting Stanford University for, I think, a workshop or something, and I showed that animation of that box folding, which was this periscope here, to Jean-Claude Latome. And he said, he looked at it and he said, geez, you know, have you ever thought about applying this to proteins? And, you know, honestly, I had never thought about it. And that was what got me started. So I went back to Texas A&M and I thought about this, and I talked with my student, and I looked up, you know, I Googled at the day, basically we didn't have Google, I guess, I don't know what it was. But I looked up to see who at Texas A&M was doing protein folding, and I just sent a cold email. I was an assistant professor, I didn't know any better, to, you know, one of our most important professors. So I sent him a message, I said, you know, I'm interested in trying to apply motion planning to protein folding. Can I come talk to you about it? And he like fired back right away and invited us over to his group meeting, and I showed him this, and I've still been working with him today. But it was all these accidental, you know, coincidences that really got this line of work going. And I, you know, encourage you to kind of think about this in your own work, to think about different, you know, things you might be able to apply the techniques that we develop in robotics community to other problems. So here's a few, just a few of the different types of problems in biology that we could apply that we could apply these methods to. The first one on the top, there's basically two different caricatures of a large protein molecule and a small drug molecule. The green is the drug molecule, and this is looking at molecule docking. So the drug molecule needs to fit into some binding site on the surface of the protein molecule. We have some protein, some small protein, animations of protein. They're not showing up too well here on the left, and RNA, which is shown at a different level of detail. So these are just some of the examples that types of problems we could study. And here's just, this is not exhaustive at all, but I just wanted to throw up the names of some of the people from the robotics community that have actually worked on problems in biology. And some of them you may be aware of, but some of them you may not be aware of, that those people that you're reading their papers in the robotics conferences, that they're also working in biology. I'll only mention really the first one, because I think this is the first one that I'm aware of where the sampling-based methods were applied to these problems. And this was an application for ligand binding, which grew out of a course project in Jean-Claude Latome's course at Stanford. The student, Singh, was taking it, and the paper that they published in ISMB, an important computational biology conference, was co-authored with his advisor, Brutleg, and Jean-Claude. And then there's many others that are doing this, so I apologize if I've left someone out, but just see that it's a lot of people that have contributed, fundamental results to our field. So now I want to talk a little bit about just the problem of protein folding, and I'll focus on that more and explain to you a little bit how we actually apply these methods to that. So first off, I want to make sure that it's clear that we're interested in the folding process. So many times when people hear or think about protein folding, what they're talking about is protein structure prediction. And that's a different problem. The protein structure prediction problem is you're given your sequence, that's just a sequence of amino acids, and you'd like to predict what the final three-dimensional structure is. That's interesting because the three-dimensional structure is related to what the function of the protein is. So if we know something about that structure, it can help us understand about the protein. But that's not the problem we are interested in. We're interested in assuming that I know typically that 3D structure. We're interested in how does a protein actually obtain that. So the folding process itself. Now there's important reasons for studying these two. This can give us insight into protein interactions and function. There are many important diseases such as Alzheimer's or mad cow disease that are related to misfolding a protein. So if we could get some insight into how a protein folds or maybe why it might misfold, this could provide biologists important tools. Now, computational techniques are really important for studying these types of problems. That's because, number one, it's hard to study experimentally because it happens very quickly. And you can't just pull out your movie camera and take a movie of the protein. There are techniques that have developed recently that allow some of this to happen, but it's still not mainstream and it's not easy to do on a large scale. And then also, if we have techniques for these, we can study many of these proteins. There's thousands of proteins where we already know their native three-dimensional structure, so we can use those to study. So, basically, hopefully you are clear that we're not doing protein structure prediction and we're trying to understand the folding process itself. And when you think about it like that, then the protein is very much like an articulated linkage, and we are interested in modeling it and applying our tools to it. So the next concept we need to understand a little bit about are folding landscapes. And these are basically, you can imagine it as configuration space augmented with energy. And the native state of the protein, that final state that it will go to, so most proteins are thought to achieve a essentially unique global minimum, a conformation that achieves the global minimum of this energy function. That's the most stable state. And conformations that are not the native state would have higher energies. So you can think of augmenting this potential energy to the configuration. And the importance of this is that different proteins will have different landscapes, and then they imply different folding behaviors. So for example, there's this kind of funnel. This is try not to be deceived by this. It looks like it's a three-dimensional state, but it's really, thousands of dimensions really. But the funnel analogy is still useful because we kind of think that it works a little bit like this, that the states that are closest to the native state are lower in energy. Now, if this landscape was kind of steep and smooth, then the folding behavior, the protein may fold rapidly and achieve the native state very quickly. Versus you may have a rough energy landscape where the protein will move around a little bit and spend some time in some intermediate conformations on its way to the native landscape. But the bottom line is, if we could understand the shape and characterize these landscapes, this could tell us something about the folding behavior of the protein. So the way that we are going to apply the probabilistic roadmap method to this problem is we're going to try to use it to essentially construct an approximate map of the folding landscape. And we are going to do it essentially, I'll go through in detail with the different steps in a bit, but we're going to do it really in a very similar way that we do for a robotics problem. And what we'd like to do is this map will be good, we'll consider it to be good if it could approximate the main features of our folding landscape. It has some advantages or some kind of nice features about it, is that once I built this map, there's actually many, typically thousands of different pathways that I have encoded in this map. I can pick any one of my configurations that's far away from the native state, and I can pull out a pathway, a trajectory from that conformation down to the native state. So for those of you that may be familiar with other methods for studying protein folding, I just wanted to try to contrast this and kind of show you where it fits in that. So there are trajectory-based methods like molecular dynamics or Monte Carlo simulation techniques. These are ones which are, you probably may have heard about. And those typically you start with some conformation and you look around the landscape kind of locally and you choose your next step and you do it again and you do it again. And when you're done with that, you have a trajectory. The quality of the path, like the resolution of this, is typically quite good, and it's under your control as to how good that may be. It can take a very long time to do that. There are, these massive distributed systems where people are applying, massive parallel processing to be able to do this to, still I would say today relatively small proteins. You don't get a map of the entire landscape at this thing, you get a trajectory. Now there are some statistics-based methods which don't give you trajectories at all, but they do give you some measure about some properties of the landscape, whether or not it's smooth or not. And that can give you some information about how quickly the protein might fold. But it's not going to give you these trajectories. These methods that we build with these sampling-based strategies, I think fit kind of in between. They are complementary to them, and ideally one would use them together with these other methods to learn more. But basically you get this map and you can have many trajectories in it. Any one of those trajectories that you pull out from this map is not going to be as good as one that you might generate from molecular dynamics. But you do get many of them rather quickly. And essentially the amount of time you spend building that map is up to the user. You can decide what level of granularity or accuracy you want. So that's just kind of to try to help you see where it fits together. Now for those of you that are familiar with the probabilistic roadmap methods, which I hope is most of you, this just lets you see kind of how the comparison goes when we want to apply this method to a protein. So in the robotics parlance, we have configuration space. In protein, I'll get into how we model it in a bit, but we have this thing called conformation space, and in some communities in biology, they actually do call it configuration space as well. But it's essentially the model of the molecule, and it models it as an articulated linkage. You need some kind of validity check. So typically in our robotics applications, that's often a collision check. Now in the protein folding application, we're looking for an energy. You need something that computes energy. And that's your basically validity check. And based on those two things, having the model and being able to generate configurations and this validity check, we can build our roadmap. And then we query, and during the query phase, we want to pull out paths. Sometimes in the original version, and kind of the simplest version of the probabilistic roadmap method, we're just simply looking for any path. The difference here is we do care about the path. We want a path that's energetically feasible, and we do that by encoding the weights into our roadmap. Okay, so now I want to go on and explain to you a little bit how we model protein. So protein is basically a sequence of amino acids, and for each amino acid, we're basically going to use in our model 2 torsional degrees of freedom, which are called phi and psi angles. So if you have a, say, relatively small protein that has like 50 or 60 residues, you're going to have 2 degrees of freedom for each residue, so you're going to have 100 dimensions in your vector that corresponds to a configuration of your protein. And we can sample a protein configuration simply by setting those torsional values. just like you could come up with the configuration of a articulated linkage by setting those angles, those joint angles. So sampling for the protein, the way that we have done this typically is we've done it where it's biased towards the known native state. And we do that because we are trying to kind of sample more densely in the space near the native state and less densely as we move out, and that's because of this kind of known funnel structure of the native state. And when we do that, we generate a sample basically by perturbing one of our existing samples. So we start with the native state, and we perturb the joint angles a little bit, and then we calculate the energy of that conformation. If the energy is low enough, we keep it. That's low beneath this E min. If it's too high, we toss it. And if it's between these thresholds, we keep it with some probability. The energy function that we used is something that's very simple. It's essentially only including very minimal information, and we coded it up ourselves. It's based on one that was proposed by Michael Levitt, back in 1983. But as you'll see later on, it's surprising how well this very simple energy function does work. It's really taking into account very simple geometric information. So the sampling, this just kind of is a little animation that shows the sampling is less dense as you move further out. So one of our challenges here is we'd like to have more physically realistic samples, but we can only afford to have a small set of samples. We want to have few samples. That's because we are going to have to process them later on. So what we've done, found to be useful, is we use this technique called rigidity analysis that's been studied by, was used by others to study molecular conformations. And basically what it does is it allows you to analyze a conformation and identify the portions of that conformation that are kind of more rigid, that have more contacts that are kind of keeping them in place, and the portions that are less rigid. And then what we do is we sample with a higher probability in the more flexible regions and with a lower probability in the rigid regions. So as an example, the helical structure, so there's a couple types of secondary structure for proteins, a helix, which is a turn, and these beta sheets. A helix is quite rigid, so if we have portions of amino acids that are in the middle of that helix, we are less likely to sample them, to move them. So here's some plots. They're called Ramachandran plots that show you the samples that we get when we do these different types of sampling. So the uniform sampling is we just uniformly sample those phi-psi angles and we don't have any bias to anything. And that's what's shown on the left. On the top row is a protein that has just 3 helices in it, and the bottom row is a protein that has a helix surrounded by 2 beta hairpins. Then the middle 2 sets are these where we've done essentially what I told you first, this Gaussian sampling. And so we're more dense around the native state. What's showing up in the plot, these kind of solid, denser regions, those are regions that are showing where the helices are or the hairpins. And then the rigidity sampling is far on the right, and you see that it's even sparser out in these regions where there's not so much structure formed. And that's basically giving us what we would like to have happen. Another thing that people that look at proteins are interested in studying is what they call these energy landscape type plots. So on the x-axis, it shows the distance from the native state. With the left, the 0 means it is a native state. And then the y-axis is the energy. And we've also colored the configurations. So this is all the configurations in the roadmap that we sampled. The darker ones, the dark blue ones, are rigid, and the red ones are more flexible. So you see that for these 3 different types of proteins, the one on the left has 3 helices, the one on the right has 3 beta hairpins, and the one in the middle is mixed. Essentially the type of structure that's in the protein seems to affect the structure. And we've done this for many different types of things, and it holds true. if you have a protein that's mostly helices, it will be kind of spread out like that, and the hairpin, the beta hair proteins are more narrow like that. So anyways, this is just sampling. We have no connection yet, so we don't have a roadmap, we just have configurations. So the next step is the connection step. So what we do is we basically try to do the same thing that we do with probabilistic roadmap methods. For each sample that we've retained, we are trying to find the k closest nodes. For the robotics applications, typically we'd use, relatively small, well, k is a constant, we use relatively small values, depending on the application, but maybe, as small as 10 or so. We found that using, values a little bit higher for the protein, but still relatively small samples, 20 or so. And we also have to decide how we determine the distance, how close are different samples to each other. So we tried using RMSD, which is this root mean squared distance that is quite popular among biologists. It actually didn't work that well for us. Then we used essentially a Euclidean-like distance in the configuration space, which worked quite well. And then we have also a method that we developed that takes into account this rigidity information. But no matter what, you need to have a distance metric to decide which are your ones you're going to connect. And then we need to assign a weight to our edges that will give us some indication about how energetically feasible that transition is. Because that's essentially what we're trying to encode here, is how likely is the protein to move from one configuration to another. And it's all about energy. So what we do is we essentially discretize at some fixed resolution the edge, and they're going to have different lengths. And we are going to compute the energy for each one of those intermediate configurations. And then we have some calculation that we do that essentially allows us to add up these values and get something that approximates the energetic feasibility. So down here, the idea is you'd like to have the lower weight means that the more favorable the transition would be. So that one on the far left there, rated 1, is almost completely downhill, so that's quite a nice one. The next one has a little bit of up, then down, then a little bit of up at the end. The last two are less favorable because there's quite a lot of rise in them. But it doesn't mean they're not going to get taken, because when we're going to pull out our trajectories, we're going to look for shortest paths. But it means that they'll be less likely to be taken if there's another alternative. So one of the biggest challenges, frankly, with working on these types of problems is how are we going to validate the work? And what are we going to do with it? So we now have built this map, which we hope approximates this energy landscape. What are we going to do with it? How are we going to convince someone that it's actually meaningful? Now we have thousands of folding pathways in there. And what we did is we chose to analyze the order in which this so-called secondary structure forms along these trajectories. And there's basically two types of secondary structure that are important. There's the helix and the beta hairpins or sheets. And so we would look at those in the final state of the protein, and we'd analyze the trajectory to see kind of what order did that secondary structure form. And then in order to validate our work, we tried to compare those, what we observed in our roadmaps from what was determined experimentally. Now, although experimentally they can't look at the entire folding trajectory, they can do experiments that will give them information about some of these things. They'll know that this portion of the structure is formed before another one. So this case study that we worked on, which turned out to be kind of perfect for us. It's some relatively small proteins, around 60 residues. There's basically 4 proteins that I show here. Protein G, and then protein L, which is another protein that has similar structure. They both have these helices in the middle and these 2 hairpins on the side. But they don't have, their sequence is quite different from each other. So that was really the first thing we tried. And then the other thing that we tried after that was these 2 mutants of protein G, which were built out of David Baker's lab at the University of Washington, where basically they took the protein G protein and they modified it with a few amino acids, just trying to modify the order in which some of the secondary structure forms. And the way that these work is in the protein G, this beta 3 hairpin forms before the beta 1 2 hairpin, and in the other ones, most of the time the beta 1 2 hairpin forms before the beta 3 4 hairpin. So we were basically trying to, process these proteins with our method and see if our analysis held the same thing. And luckily, it did, actually. It was pretty amazing. Even using, if you remember, I told you we had this extremely simple energy function. With that, we were still able to discern the difference between these two different, these different folded behaviors. Basically, when we were looking at it, for protein G, about 99% of our paths had the correct formation order, and in protein L, all of them did. Then when we moved to the mutant proteins, NuG1 and NuG2, we had extremely high results there, with 97 or 98% of the pathways following the same order. This is just another look at that same data, but it's just to show you that we do get some variance. We get a few of the pathways, like 0.8% or something, where like in NuG1, where the pathways form, the hairpins form in the other order. And there's several different types of experiment that have established these orders. So we were very pleased with this, and it basically kind of gave us some standing to kind of go back and say we have something here. Another thing that we can look at is when we look at the rigidity analysis, this rigidity analysis of the protein conformations, it told us something interesting. So here's the same 4 proteins that we looked at before, and we built these things that underneath there, I don't want to go into too much detail, but essentially they're showing their contact maps, and they show us, for example, these regions that are circled correspond to the beta 3-4 hairpin area, and if it's black, it's more rigid, and if it's green, it has some more flexibility in it. So you can see when I look at those same 2 regions for NuG1 and NuG2 that there's more flexibility into that portion of the conformation than there is in protein G, the original one. Similarly, when I go and I look at the other hairpin, we have the reverse situation, where the NuG1 and NuG2 are more black, so they're more rigid, and the protein G is less rigid. So even just this rigidity analysis is something that I think is, at least some of our collaborators they've just wanted us to analyze some of their conformations for them, because they found that to be somewhat interesting. So far, we've essentially built these roadmaps, and this is kind of the proof that shows us, at least that there's something there, that we're not completely, pulling it out of the hat. But there's other things that they're interested in understanding about how proteins fold, and these are things that are basically related to rates or kinetics. So you might be interested in asking questions like, which protein folds faster? Which mutations of the protein might speed up or slow down folding? Population kinetics. How does the population of a particular conformation change over time? Are there conformations that are like bottleneck conformations? So how these values have been these properties have been studied in the past there's a couple we looked at to see how they had been done. And one of the things was this master equation type method. And basically the master equation is something that's usually applied to a complete energy landscape, which we don't have, and we can't afford to build actually for anything but a very small protein. So what we basically did is we kind of reinterpreted this whole computation, and we put it on this roadmap. So we kind of assume that the roadmap is our complete energy landscape, and we apply this to it. Another type of simulation that is done on energy landscapes are Monte Carlo simulations. And here it's a similar thing, but every time step you want to compute your local landscape and decide which way to step. So what we did is we did this just on our roadmap. And then we can use that to go forward. So it's basically, substituting the complete landscape with the roadmap underneath. And others have done this as well. So this shows, so basically the second eigenvalue is something that's related to the rate of folding. And this shows here, the blue is the original protein G, and then the green and the red ones are the two mutants. And it basically shows that there's a big difference in our calculation as to the folding rates of these, and it did correspond to what's known in experiment. Another thing here, this is a little bit harder to see from these plots, but we were looking at the population kinetics. How does the population of different conformations evolve over time? And we also found that we agreed with what's known for that over time. So we found the ones on the right correspond to new G1 and new G2, and you can see that at the initial part of the plot there, they are accumulating faster, the black part. The folded state's the black part, and the unfolded state's the purple part. So it's basically confirming that using this approximate landscape it's not going to allow you to get the rates precisely, but it's going to allow you to come up with relative information. That's just time here. So one last thing I want to share with you is something that one of my students who's now at the University of New Mexico is working with, and it's a kind of an interesting application. She organized a workshop here a few days ago with some others. So basically here, she's studying allergies, and basically how things work is that allergens basically interact with the cell surface and form aggregates. And the shape and the structure of those aggregates is thought to indicate the allergic response severity. So a lot of people have allergies, probably many of us here. And some of them are so severe that people actually die from them. So like shrimp or peanut allergies are some very serious ones. So what they're trying to do is they're trying to apply motion planning-based solution to some of these problems. So they start with essentially an all-atom structure, which is shown in A here. And then they generate this molecular isosurface using known techniques. And then they try to simplify that to get to reduce the geometric complexity. So they go down for something that has roughly 350,000 polygons to something that has maybe 1,200 polygons. And let's see, I think this will play on its own. So this was a simulation, yes, okay, where they have used motion planning-based random sampling, so similar sampling that we've indicated there, to kind of guide the simulation. And there's experimentally derived association and dissociation rates and molecule speeds. So those things are kind of put into the simulation to make it fit. And then they one thing that's known and that showed up in their simulation properly too is that the aggregates basically move inversely proportional to their size. Not that surprising. But she is working with some collaborators there at the University of New Mexico, and they're further trying to refine some of these techniques. And they have also developed a haptic game for doing molecular assembly. This is Lydia with her daughter playing with it. You can kind of see they're moving the orange part around in the site there, and they're getting the force feedback to show them whether or not if it's fitting well into the pocket. Okay, well, it looks like I finished well ahead of time. So what I hopefully have convinced you of is that sampling-based motion planning can be used to model molecular motions. And really the key, the only difference that we had to do is we have the same exact code that we use for all of our robotics and other applications, and we apply it to these problems. What we have to do is we have to come up with the model for the configuration space, and so that does require that you develop that individually. But once we did that for a protein, we can apply it to any protein. And then the validation check. This is something which you need to develop, and we did essentially almost all of our work uses this coarse energy function that we developed based on excuse me, Michael Levitt's work. We did also compare against an all-atom energy model, and essentially what it did is it just kind of made our results stronger, but made us take a lot longer to compute them. So for example, something that would take us an hour, say, on one of our systems might take 2 weeks if we moved and used the all-atom energy function. But the results, kind of the relative information that we discovered, were no different. We have some results that you can browse on this folding server on our website. And I'd like to also point out and thank some of my former students. Some of them have gone on and are doing other things, and some of them are continuing to work in these areas with their own students. And then my current students, two of who are here, Chinwei and Cindy, are here today. And also my colleagues that are listed on the bottom, those are the key to doing this work. You can't do this kind of work without good collaborators from the discipline. And so I have, most of my collaborators are from biochemistry. So with that, I'm happy to take any questions.
Do we have questions from the audience? Exactly.
Hi, how do you push the domain knowledge from this area into the algorithm, the motion transit algorithm? Like what techniques do you use?
Well, what techniques? I'm not quite sure I understand your question, but like what we did for the domain knowledge, I guess, you can tell me if I'm answering your question. The rigidity theory is something that we ended up using to analyze the conformations, because we knew that there was some parts of the conformation that would be, it just wasn't physically realistic to perturb any part of the configuration randomly in the sampling process. So we used the rigidity analysis to determine what parts were more rigid and which were less rigid, and then tried to perturb them based on that. And that information we basically developed together with our collaborators.
Hi, great talk. I was wondering if you have used your methods to understand at all proteins that change their conformal structure, for example, hemoglobin or other proteins that change their structure when they bind to something else.
Well, actually, that's one of the things we've been trying to make progress on, but haven't yet. So I think you're talking about things like also allostery is a type of reaction where if you have like something binds with a protein in one part of the protein, it can cause conformational change or structural change in another part of the protein. So there's, trying to understand. I think that we should be able to actually use the rigidity information to kind of see how structure accumulates and transfers through the protein.
Along the same lines, instead of have you thought about extending not only from analyzing the roadmap, but actually extending it to control? What if you wanted to try and control the protein to fold in a certain way? Are there ways of introducing other molecules that would help guide the folding?
Yeah, great question. So everything I talked about today was basically looking at a protein all by itself. There was nothing else there really. And so this would be something like, is there something that we can put in, another structure, or even the ligand or protein or something? And that's another thing that we're kind of, I would say, in the initial stages of working on. But another thing that people are interested in, and we have done some work with, is what happens when you actually mutate a protein in a particular way. How does that change things? And the challenge there is all of our work, we have been assuming that we knew the native structure because that had been determined experimentally. But if somebody wants to mutate a protein, we would like to do this, without having them actually do it. So it's a challenge for us to do that, and we need to try to come up with good strategies for kind of convincing ourselves that we do understand what that final state is. These new G1 and new G2 proteins give us, some confidence that we are kind of not too far, especially if the mutation is small enough.
For this very interesting talk, I was wondering what's missing from motion planning methods to try to do protein structure prediction?
Well,
if something is missing.
it's basically the energy function, right? So one of my collaborators actually thinks we should be able to, he's still convinced, Kendall, he was my first collaborator, and he's still absolutely convinced that we should be able to do protein structure prediction with this. And he thinks that we can use these techniques to help us learn and improve the energy function. And we have done that a little bit with him, but basically, if you know the energy function, then it tells you which way to move. But otherwise, you know, where it's not, what I showed you today is not doing structure prediction because it's mapping the landscape, right? If we can come up with a good map of the landscape, we should be able to do protein structure prediction as well. But can we do that without having information at all about the native state? Because we're using the native state in all of our work.
All right. Yeah, so I'm just wondering, it's kind of surprising that these methods are able to use sampling in a high-dimensional space effectively, you know, despite the fact that the energy landscape is, you know, might be very complex. So do you think that's really saying that the energy function is actually more structured than you might believe otherwise? Like maybe in a probabilistic sense, say, because the energy function is composed of a number of independent components, that there's actually some probabilistic convergence to a more structured object, or is there any theoretical work trying to analyze that aspect?
Well, I mean, one of the great strengths of these sampling-based methods is you can kind of apply them to a very high-dimensional space. If they're going to work, you've basically hit the nail on the head. You need to have some structure in your underlying landscape. You know, you need to have something to map, right? And if you are going to map it with this very sparse set in a very high-dimensional space, if the map will be meaningful, you do need there to be structure underneath it. Ken Dill, you know, the colleague who I mentioned before, one thing that always bothers me is the way that we are weighting our edges. So we basically take the two endpoints of the edge, and we take them at some fixed resolution, which we decide a priori. All edges have the same resolution, and we compute these intermediate, you know, that we analyze each one of those, and we just say that that's an approximation of the edge. But the edge could be very long, and the landscape between those two edge points might really be something like this, and I've approximated it with this straight line. And it bothers me. I still today, I'm, you know, not convinced this is a good thing, but, you know, he's basically convinced that there is this underlying structure, and it's a good approximation. And, you know, even if it's, you know, if it's too long, it's probably not, but if it's out there in the space of the landscape, that it's a longer space and we don't really know very much, you know, maybe it's okay. And, you know, that's his intuition. You know, he basically believes what you're saying. I defer to him.
So do you think that any other motion planning techniques, and basically I'm talking about potential function-based motion planning techniques, would have any luck in tackling this problem?
In methods besides the sampling-based methods? Well, I think the sampling-based methods are best suited. You know, if you think about it, ours is kind of maybe a hybrid between a tree-based method and a PRM or graph-based method, because we are starting from some known states, and we're also doing things where we're studying transitions between we know different conformations, so we'll maybe seed our roadmaps from several different points. But I think that this high-dimensional space makes it very ideally suited for the sampling-based methods. And I just believe that they're good methods. I haven't honestly thought about if other methods would be well-suited. So I don't know. Do you have some idea of one that you think is? Yeah, maybe later.
Could you give us a sense on how much impact these robotics techniques had on computational biology in a broader sense, are they now becoming kind of mainstream? Are they well accepted?
Well, not as much as we would have liked, I would say. But I think that one of the challenges is that much of the work in biology is still very sequence-based. They're not really using the three-dimensional structure as much. And I think that, you know, we have a lot to offer in understanding like the geometry and the spatial information. So, you know, I think that some of the forward-looking people in these fields are, you know, paying attention, and they like to collaborate with us. So, you know, I think that we're not there yet for sure. But there are a lot of people that have heard about it. So it's rare that I, you know, meet someone who's like completely, you know, not aware of these different types of approaches. Usually they know about it from the collaborators in their own discipline. So, you know, I don't know. Maybe Lydia has an opinion on that.
Let's thank Nancy.
Let's see.